is. Do you know who we can interview?
I have no idea.
Guys, I know who we can interview.
Who?
There she was, just a walking down the street, singing do-a-diddy, diddy-dum-diddy-do.
There she was, just a walking down the street singing do-a-diddy-diddy-dum-diddy-do, snapping her fingers and shuffling her feet singing do-a-diddy-diddy-dum-diddy-do.
I'm Natalia. I'm an assistant professor in the Department of Agronomy, Plant Breeding and Plant Genetics program. The Plant Breeding and Plant Genetics program is a program that encompasses several departments on campus, and we all focus on different aspects of improving plants for either food consumption or other uses in agriculture. Well, I am originally from Uruguay, South America, and I studied my degree in agronomy at the University of São Paulo in Brazil. And then I moved to Argentina because of my family and graduated in Argentina and in agronomy. And since very early on, I knew that I wanted to work in something that will relate agriculture, food production, and people. And so plant breeding seemed to be the thing that really really completed that desire, that you are involved with food production, but also food production in the context of a community, and the community could be a city, a country, or the whole world. So that's how I decided for that career. And when I finished my undergraduate in Argentina, I came to the U.S. to do grad school. I always knew that I wanted to continue my studies, and I did visit a lot of universities, but I was lucky enough to find a position actually here at the University of Wisconsin. So this is where I got my master's and PhD in this same program, Plant Breeding, Plant Genetics. So our research is focused, we work with maize, with corn, and my predecessor, the person that was in my position before I started, he worked in improving corn for silage purposes. Living in Wisconsin, the dairy land, milk production is very important, so feeding these cows is a major priority for agriculture in the state. And so he started selecting for improving composition, the quality of corn and yield for silage purposes. And a lot of the research that we do revolves around that. It's trying to understand how we can make these plants more productive and better suited for ruminant nutrition. And then about 5-6 years ago, in 2007, the University of Wisconsin received a fairly large grant from the Department of Energy to study how to utilize plants or vegetative material for the production of biofuels. And we got involved in that project from the very beginning. And the reason why we got involved is because we knew how to look at plants as a source of forage. You know, if you think about the rumen of a cow is a big fermentation chamber, and these biorefineries that people are intending to develop to produce biofuels are just large fermentation chambers. So all the knowledge and all the germplasm and all the information that we accumulated in our work looking at forages kind of fitted right into these other projects. So we've been working on aspects of how to utilize plants for the production of liquid fuels primarily. Well, a lot of what we do is we look at natural variation, primarily in corn, and evaluate that diversity, and then look at, you know, for specific traits. In the simplest way, we say, okay, if we want to select for plant height, we look at a population and take diversity from the lowest to the tallest plants, and we put them out in the field, and we look at those plants in the field and measure plant height. And then we take, we use genomic information, particularly genomic markers, DNA markers or RNA markers, and then look at the same type of diversity at the DNA level or RNA level, and then try to associate variation in the phenotype with variation in the genotype to try to identify, through associations, we can identify regions of the genome that might be related to the traits that we're interested in. And the idea with that is that if we know something about how the genome operates and what are the main players that are contributing to a certain trait, we can more easily alter that genetic structure to improve plants. So we do that sort of as a screening method to identify genes, identify regions figure out more efficient ways of selecting plants for the purposes. But then if we identify genes that are really interesting, we could use genetic transformation to improve the particular plants. In our setup, we mostly use it for research purposes, to understand how those genes behave in certain backgrounds, and so we can understand the biology. We do have a breeding program per se, where we improve populations, we release what we call inbred lines, or that could be transformed into hybrids, and people could have access to that. People in industry, public sector, or private or public sector. But that process of breeding does not involve any genetic transformation. All of that is purely based on phenotype, because it is our choice. It could fairly well incorporate some of those other technologies. But we do use genetic transformation and a lot of genomic tools for the research aspect of it, where we are trying to understand how we can more efficiently improve these plants. On Wisconsin.